Okay, welcome back everybody, and thank you for joining us again for the second half of the showcase. It's a pleasure for me now to welcome Professor Sergi Castellano, who is the Deputy Theme Lead of our Genomics and Systems Medicine theme within the BRC. And Sergi will be chairing the next session. Thank you, Sergi.
Thank you, Laura, for the kind introduction. I will be very brief for the sake of the session and the speakers, but I'd like to mention that we're going to move into the use of genomics for the investigation of rare disease, but most importantly, for the rapid implementation of these technologies for the diagnostics and new treatments for the children under NHS care. Now, our first speaker is the lead of the theme and also professor of genetics and fetal medicine, Lynn Chitty, who will give us an overview of some of the genomics work that we have been doing in the last few years. Thank you, Lynn. If you can share your screen.
Thank you. Can you see that?
Yes, that's fine, Lynn.
Okay. Okay, thanks very much, Sergi. So I'm going to talk to you about genomics and systems medicine. And, well, I'm not, because the slides won't move.
Try pressing your mouse button once, Lynn.
No, the mouse won't do it. The mouse is, okay. So the overarching themes of our theme, aims of our theme, were basically around gene discovery, delivering multi-omic pipelines and establishing rapid sequencing, and also extending the use of prenatal diagnosis using cell-free DNA. Over time, that's evolved to include single-cell analysis, exploring different applications of cell-free DNA, and using long-read sequencing for gene identification, and also responding to the pandemic by supporting pathogen genomics sequencing. So in terms of gene discovery, our theme has, over the last two BRCs, had GOS-gene, which identified a number of genes, but there were many still left undiagnosed. With our reanalysis using short-read sequencing and other approaches, we've actually identified in this cohort many new genes, 20 new genes, new mutations, and some associated phenotypes across a wide range of conditions. We've also capitalized on the many over 9,000 rare disease probands that we recruited in the 100,000 Genome Project. And with a BRC-funded postdoctoral fellow working jointly with Moorfields and BRC-funded PhD students and clinical fellows, we have identified the diagnosis in more than 100 undiagnosed cases. And we've now recently extended our approach to diagnosis using long-read sequencing for other conditions as well. And I'll explain how we're going to continue with that. We're also leading the national evaluation and the economic evaluation of the return of additional findings from the 100,000 Genome Project. And that's a project which is ongoing at the moment, and the return of these results will finish in June this year, and it'll take us another few months to evaluate health professional and patient experiences of this to inform policy for sequencing in the NHS. The other thing that we did with capitalizing on 100,000 Genome Project are two other projects. One was whole genome sequencing is complicated, and we developed two short infographics explaining whole genome sequencing. And we developed these with YPAG and local school children, and they've subsequently been translated into 4 languages. And the original videos were actually voiced over by a member of YPAG. As you can see here, they've had many thousands of hits on YouTube, and they've also been viewed very highly on Chinese social media. We've evaluated the value of these and shown that children and their parents like the format and helped understanding. And my personal experience with my family, older children and adults, is that they also help adults understanding too. In addition, we've worked with patients and lay support groups to develop a measure of informed choice for whole genome sequencing and to ensure that we are appropriately offering this to patients. We have developed and refined pipelines and validated them pipelines for transcriptomic analysis in many situations. And these are just two examples here where we've actually managed to identify, in the first instance here, the transcriptomic profile of subphenotypes of juvenile dermatomyositis to identify the subgroups that can be used for targeted therapy, so personalized therapy, by identifying these subgroups. And we've done a similar thing to identify subgroups of patients that will relapse with ACP. And this then allows us to predict who's going to relapse and inform further therapy. These are just two examples, but there are many more ongoing. One of the things that we have done and is proving very successful is the development of single-cell transcriptomics and proteomics. And we've also established the computational pipelines for these single-cell analyses. And this is very much in collaboration with Gosh Children's Charity. So one of the things that this is enabling us is enabling us to improve therapies for skin diseases and also for leukemias. But I'm not going to say any more because Dorian, our BRC funded research fellow, will discuss this in more detail later. But what we are also doing, and I think this is quite exciting being a fetal medicine doctor, we are classifying and concentrating amniotic fluid IPCSs, both for postnatal, potentially for postnatal treatment and also in utero therapy. We have established rapid paediatric sequencing. Establishing a genetic diagnosis in these critically ill children is really hard because of the genetic and phenotypic heterogeneity that can underlie their conditions. We developed a pipeline for rapid whole genome sequencing and tested this in 39 trios, identifying the causative variant in 35% of cases in 10 days. And this then allows you to inform management and treatment. And what have we done subsequent to that? Well, just a little sideline here. In 2018, the NHS established the Genomic Medicine Service to deliver personalised medicine. This genomic medicine service is delivered from 7 genomic lab hubs. So laboratories have been consolidated into 7 lab hubs. And these laboratory hubs deliver testing based on a national genomic test directory. And this is to enable equity of access to all patients across the whole of England. Our Gosh leads the North Thames Genomic Lab Hub, and Birmingham Women's and Children leads the Central and South Genomic Lab Hubs. And these are the two of the biggest lab hubs, and together we serve half the country for specialist testing because of the range of specialist tests that we offer in our GLHs. So I just want you to remember about the GLHs, because I think, and the Genomic Medicine Service, because it is important to how we've translated our work. So the work I've just described about rapid paediatric sequencing was subsequently adopted to the National Genomic Test Directory in October 2020. And this service is currently delivered by the Southwest Genomic Laboratory Hub. And an analysis of the first year of work showed that the 39% of the 364 babies referred for diagnosis received a genetic diagnosis. The average turnaround time was 14 days. And diagnosis changed management in around 90% of cases. The referral rate has nearly doubled since the first year, and there are now around 700 babies a year referred for testing. So this work is now impacting families across the country. At Gosh in 2021, working with the BRC researchers and lay support groups, we started the evaluation of the service, and we have subsequently achieved research for patient benefit funding to evaluate the service implementation across the country. So looking at equity of access, health economics, and patient and health professional views. And you can see from these two quotes here that both the parents and the genetic counsellors and the health professionals delivering the service find it's a very positive service, and we have a lot of very positive feedback. And but we want to establish rapid whole genome sequencing as a clinical service in our GLH at Great Ormond Street, so that we can test these patients in-house in an accredited pipeline. Whole genome sequencing is now available through the NHS Genomic Medicine Service, but the turnaround time is too slow for some indications, one of which is early onset epilepsy, which Amy McTeague will talk to you about later. So together, the BRC and clinical bioinformaticians and scientists have developed an in-house rapid whole genome sequencing pipeline, which is now functioning and offering turnaround times for whole genome sequencing in a very short period of time. We plan to work with NHS England to bring other tests beyond the early onset epilepsy that Amy will talk to you about in-house. So, for example, the immunodeficiencies and very sick neonates. We also plan to develop pipelines to rapidly investigate the tier 3 and unsolved cases and translate this to clinical practice, because there is still a huge burden of unsolved diagnoses even following whole genome sequencing. We will then work with our partner trusts, Birmingham in particular, to deliver this more widely across the NHS. So to continue on the theme of rapid sequencing, we've also developed rapid sequencing for the diagnosis of the dysmorphic fetus. So 20% of pregnancies are complicated by unexpected fetal abnormalities. And whilst chromosomal and other tests can identify the underlying etiology in around 60%, there's still a good 40 to 50% that remain undiagnosed. But we know that exome sequencing can improve the diagnosis in this cohort. So we developed a rapid pipeline to analyze these trios. Then we do trios, we test mum, dad, and the fetus and sequence them rapidly. And our work showed that you could do this and turn it around in 10 days. So this is the pipeline here, from the detection of abnormalities, receipt of the sample in the laboratory, to issuing the report. We did this in an average of 10 days for about 40 fetuses. We use this data, and we also work with Birmingham, our partners in Birmingham, to identify cases most likely to have a genetic etiology. And this is the results from the PAGE study, which was published in The Lancet in 2019. And here you can see that skeletal abnormalities in fetuses, hydrops, or multi-system abnormalities have the highest diagnostic yield for exome sequencing. Using these data, we need to identify which are going to be the most likely cases, because this is expensive. It costs about £2,500 per trio to run this at the moment. Anyway, using this information, NHS England decided to implement whole exome sequencing service for fetuses with structural abnormalities in the NHS Genomic Medicine Service. The service is delivered from Great Ormond Street, North Thames, and Birmingham Central and South GLHs. In the first year of the audit, which we're just starting now, we diagnosed 116 of the cases sequenced, that's 35%. And I'm very pleased to say, if you look at this heat map here, that the diagnosis, the referrals came from all over the country. And remember, we're just testing for England at the moment, not the devolved nations. We now have a BRC funded clinical fellow who is doing a PhD looking at the implementation, and we also awarded HS&DR funding to evaluate the implementation of this work. The study is called Express. Again, early studies show that both patients and health professionals feel that this is a very positive service, but we are identifying some issues. For example, this patient really felt it was very overwhelming, the discussion of what whole exome sequencing is, and would need more time to go over things. And it's clear that there's a big educational piece that needs to be done, particularly in fetal medicine and midwifery. We've expanded the applications of circulating cell-free DNA. So our work over the years in the GOSH BRC has enabled NIPD, non-invasive prenatal diagnosis for monogenic disorders, based on a maternal blood sample to be delivered. And this too has been implemented in the National Genomic Test Directory. It's delivered from North Thames and Central and South, and currently about 300 families a year are benefiting from this service. We are at GOSH trying to expand the application by exploring droplet digital PCR for sickle cell, and also for maternally inherited variants. Maternally inherited variants are very difficult to detect because there's a high background in mum's blood of her own mutant allele. And we're also beginning to use long-read sequencing for parental haplotyping to increase the availability for parents carrying recessive diseases. In the course of this BRC, we have worked to develop donor-derived analysis of circulating cell-free DNA for monitoring graft health. So here you can see, because the DNA from the donor organ is different from the recipient, we can analyze the donor DNA in the blood of the recipient. And here you can see, for this renal case here, high levels of donor-derived cell-free DNA immediately after transplant, which falls rapidly, and there were no problems here. However, in this case with the heart transplant, you can see that as the rejection episode began, the level of donor-derived cell-free DNA increased. And we hope to be able to use this to monitor the graft health and avoid needing to use invasive biopsies. But that work is ongoing and hopefully will continue in the next BRC. And finally, we responded to the pandemic by developing COVID-19 genomics. We contributed to the COG UK study led by Judy Brewer in London. We sequenced more than 20,000 COVID-19 genomes to monitor the spread of the virus and the variants of interest. We've also contributed to the COG UK hospital onset COVID-19 infection study to enable rapid tracking of viral spread in NHS hospitals. And subsequent to our success, NHS England have invested in Great Ormond Street as one of the centres to be ready to sequence new outbreaks. And we also, we've been part of a number of high impact factor papers. So that's just a really brief overview of the work that we're doing in genomics and systems medicine, but just a summary here of how we've translated for patient benefit. More than 150 families have had received a diagnosis that explains their condition, enabling reproductive choices and new therapy in some cases. We've developed our transcriptomic and single cell pipelines to better delineate disease and target therapies. And we've incorporated into the National Genomic Test Directory more non-invasive prenatal diagnosis, rapid sequencing for the diagnosis of monogenic conditions for very sick children, benefiting around 700 families a year now. And we've also got recently introduced rapid sequencing for the diagnosis of monogenic conditions of fetuses, enabling better management and informed parental choices. At the moment, we're getting about 400 cases a year referred, but those numbers are increasing. And finally, COVID sequencing for rapid response has enabled us to respond to the pandemic and will enable us to continue to do so. Throughout, we've worked with patients, lay support groups and healthcare professionals, both to direct the implementation of some of this exciting work, but also then to evaluate the implementation and ensure development of service in accordance with patient need. And thank you very much. That's all I have to say.
Thank you, Lynn, for this wide overview. We are a bit over time, so we're going to move quickly to the next talk. So if I can ask you to stop the video, and then I'm going to introduce, as you mentioned, Dorian Chenakis, who is a research fellow in the theme, and our in-house single cell specialist who will describe some of his translational efforts. Thank you, Dorian.
Thank you. Just share my screen. Can you see my slides?
Yes, that's fine.
Lovely. Thank you very much. So yeah, thank you very much. And hello, everybody. My name is Theodoros Xenakis, and I'm a research fellow in single cell genomics, and my post here is funded by the BRC. And today I wanted to talk about the use of some cutting-edge genomic technologies and how these are being applied in a translational setting. So one of the questions might be, why do we need single cell resolution? And when we think about biological samples, such as biopsies, whether these are blood tissue or liquid biopsies, or even a simple blood sample, these are complex and heterogeneous structures. And traditionally, these techniques homogenize these structures made up of various different cell types. And therefore, the readout is a generalized average, which can dilute certain signals and markers, which can be very important. But you also lose the capacity to look at the cellular dynamics. And when you think of fields such as cancer, knowing how a specific malignant cell behaves in its microenvironment and how that interacts with healthy tissues, this can be incredibly important. So we use the 10x genomic solutions where single cells are isolated in these nano droplets along with gel beads, which are coated in molecules that are able to capture and uniquely tag RNA. And this allows us to assign every RNA molecule to its cell of origin. And now we can also treat these cells before isolating them with specialized antibodies. And this allows us to simultaneously obtain surface protein information alongside the gene expression information. And this is one of the things that takes us from single-cell transcriptomics to single-cell multiomics. And colleagues within the BRC are also optimizing the latest solutions in bioinformatics so that we can make the most out of the data produced. Now, this technology can be applied across the spectrum. So from exploratory science through to projects aiming to understand disease mechanism and all the way into the clinic used within therapeutic solutions. And what is great about the GOSH BRC is our access to very rare and very precious pediatric cohorts, as well as in our collaborations being able to draw from excellent clinical expertise to truly explore the translational power of this technology. And now I just want to talk about two projects that I've had the pleasure to be involved in and collaborate with. And these are jointly supported by the BRC and other funders, and these are ongoing. So the first area of application I wanted to discuss is in skin disease, and in particular congenital ichthyosis, which is a group of monogenic skin disorders that can range in severity. Now, the general characteristics, as you can see, are thick, rough skin that is frequently inflamed and prone to infections. And this can affect the quality of life and in severe cases even be life-threatening. Now, unfortunately, new treatments tend to be mostly palliative and can be very expensive. So here at the ICH, clinical trials were completed successfully in a project where patient skin cells were harvested and the disease-driving mutation was corrected by gene modification. And then these corrected cells were then cultured and skin sheets were grown in the lab that were then grafted back onto the patient. Now, this is very promising. However, there are a couple of hurdles to overcome. The first is it takes quite a long time to actually culture these cells and generate the corrected grafts. But also quantifying the stem-like cells in these skin sheets is very important to assess their ability to self-renew. And this translates into long-lived therapy. And for translational purpose, current methods of quantifying these cells are impractical. So here is an experiment that addresses the first hurdle done by my colleagues. ROCK inhibitor was used in the early stages of stem cell culture, and this results in a 5-fold acceleration of the process compared to a control. And for this, I also supported the application of single-cell techniques whose high resolution allowed us to establish that we can maintain normal cell properties and that skin sheets derived from these cells are suitable for grafting without us having introduced any unwanted properties. But this has also provided some preliminary results suggesting that single-cell sequencing combined with some advanced bioinformatics allow us to quantify the stem cells in culture. As you can see here, these holoclone-forming cells are the more stem-like. And as we mentioned before, this is important for long-lived therapy. And we can do this in under a week, which is much faster compared to traditional methods. So the second field I wanted to talk about is in leukemia, which is something in its many forms we have a keen interest here. And here I'm just showing the process by which blood cells mature from stem cells. And now leukemia occurs, as many of you already know, due to certain mutations in these stem cells, which then give rise to malignant mature cells, which form the bulk of the leukemia. Now, most therapies at the moment mainly target these highly proliferative mature malignant cells, whereas the malignant stem cells are much harder to target because of their quiescent nature. And it is therefore these malignant stem cells that frequently are responsible for residual disease and remission and eventually lead to relapse. And to make things more complicated, they also frequently become more heterogeneous, making them even harder to target therapeutically. One of the more promising solutions for this is CAR T cell therapy, which stands for chimeric antigen receptor T cell therapy, where a patient's own immune system can be customized to treat cancer. So briefly, T cells from a patient are harvested and are customized by introducing DNA that codes for a chimeric antigen receptor. And these receptors are specifically designed to target proteins that are specific to cancer cells. These CAR T cells are then expanded and infused back into the patient where they can target and destroy malignant cells. And as mentioned previously, earlier on today, here at the ICH and the BRC, there is track record of using CAR T cells in pediatric leukemia. So one very important element of this therapy is identifying proteins that are present on the surface of malignant cells that are specific to the cancer and that these chimeric antigen receptors can target. Therefore, as you can imagine, being able from a diagnostic sample to identify these stem cells, distinguish the malignant stem cells from the healthy ones, and at the same time interrogate the protein landscape on the surface of these cells would be incredibly powerful. And that is exactly what we have done. So I supported a single cell experiment in collaboration with people from the Giustacchini lab, which generated this data from a chronic myeloid leukemia patient treated here at Gosh with tyrosine kinase inhibitor. And we have samples from diagnosis, and remission. And here the color essentially indicates the level of stemness for each cells. So here you have the more stem cell-like, and here you have more mature cells. And you might ask how we know which cells are cancerous. And we are looking at three main approaches for this. In cases such as this one, which we know is caused by fusion of two different genes, we can look at the expression ratio of the genes involved to identify malignant cells. For use in less straightforward cases, however, we have also successfully used an enrichment strategy to sequence the actual mutation point. And finally, we are working with Oxford Nanopore to combine single-cell sequencing with another powerful and cutting-edge technique, which is long-read sequencing, to be able to interrogate both the gene expression and the mutation genotype for each cell from one data set. And we have already generated 2 such data sets, and my colleagues are busy at the moment analyzing this data as we speak. So at the same time, and for these cells, we were able to interrogate over 137 surface proteins to explore the potential for finding targets for CAR T cell therapy. Now, finally, apart from offering incredible insights into therapeutic techniques, being able to interrogate diagnostic and remission samples at such a resolution also offers a very personalized prognostic approach by being able to assess the presence of malignant stem cells that are evading initial treatment. And with that, I just would like to thank all my colleagues and all the patients who have been involved in the collaborations. And here I mentioned 2 great examples of pediatric disease where these technologies have been able to be applied translationally. However, these methods really are truly universal and can be applied in the context of any disease, really. And just on a personal note, I've really enjoyed being part of the BRC's effort to use exciting and novel techniques and technologies to improve outcomes for pediatric patients. And it's been a great opportunity for me to further develop and apply my expertise in a translational setting. Thank you very much.
Thank you, Dorian, for this clear exposition of these advanced techniques. I understand that we have a couple of questions. Back to you, Laura.
Oh, yes, we can take them now. There's one question for Lynn, if she'd like to rejoin us. Lynn, there's a question on Slido that says, what 4 languages were the whole genome sequencing infographics translated into?
2 Chinese, dialects, and Turkish and Bengali.
Great, thank you very much. And then one slightly more open question that said, how does a research group go about collaborating with the genomics groups? They're talking about capacity potential limitations. So how do we manage that?
Well, they just need to approach us and discuss what it is they want to collaborate on, and we can work out whether we have capacity or not, or how we're going to go about funding. So
okay,
we're
that's
very
great.
happy to take questions or take ideas from anybody.
Good to know. Thank you. Okay, Sergi, I think we can move on to the next talk.
Yes, thank you. So our next talk, which is recorded, is by Amy McTagg, who is one of the junior faculty colleagues, now also an MRC fellow and a consultant on pediatric neurology. And she will talk to us about the use of genomics on children's epilepsy. So if we can start the recording, please.
Hello, my name is Amy McTagg. I'm a research fellow and consultant paediatric neurologist at the UCL Great Ormond Street Institute of Child Health. I'm going to talk to you about epilepsy in children from bedside to bench and back again. So the journey from being a clinician to a clinician scientist is one that I've taken through epilepsy. And the reason I do this is because 1 in 1,000 babies in the UK under 12 months of age are affected by epilepsy. Unfortunately, 50% of these babies will have a genetic cause for their epilepsy. And although this is fantastic, we can make more diagnoses nowadays, we still have a real lack of effective treatments for these children, and many of them will have frequent seizures that do not respond to treatment. Unfortunately, as well as frequent seizures, these children also have very poor developmental outcomes. So there are a range of impacts, from seizures to poor developmental outcomes to increased mortality, and big effects on parents and families and the wider society. So therefore, as a developing clinician scientist, I'm aiming to use my earlier gene discovery, to use this to understand disease mechanisms in the early onset epilepsies, and then to use this knowledge to develop novel therapeutic targets which can be used as novel therapies for patients with epilepsy. So we know that there are many different genetic disorders that can underpin the early onset epilepsies, and in my earlier work I identified some of these and understood the range of features that these can cause. And you can see here in the top of the slide some sodium and potassium channels and a potassium chloride co-transporter. But I realised by the end of my PhD in my early postdoctoral period that what I really wanted to do was to understand in a patient-derived model the direct impact of those mutations on neuronal function and to understand how these mutations lead to epilepsy in a bid to try to get to better treatment. So therefore, I'm now moving to the bench to look at a patient-derived model of early onset epilepsy in my MRC fellowship, which was supported by having obtained the earlier BRC Catalyst fellowship. So I've taken skin cells from my patients here at Great Ormond Street. And the reason that I want to look at a patient-derived model is to better understand the impact of the mutations in the patient's genetic background. And working at Great Ormond Street gives me the opportunity to meet and look after many patients with rare diseases, and therefore to obtain these patient-derived samples. These have now been converted into pluripotent stem cells, which have the ability to form any form of cell in the body. And I'm converting these into cerebral organoids, which are small collections of neurons, almost like mini brains, with other forms of organoids which contain inhibitory neurons, and fusing these into assembloids, allowing us to look at the interaction between excitatory and inhibitory neurons. And we've already shown by validating the pipeline that we get increased expression of key developmental genes such as KCC2 throughout maturation of the organoids. We're now using phenotypic assays, so looking at what these organoids do in culture, and looking at both electrical networks using electrophysiology, but also looking at gene networks using single-cell RNA sequencing in collaboration with the genomics team. And this is really to look at a single-cell level and what the differences are in gene expression between mutant lines, control lines, and gene-edited lines where we've corrected the mutation. But the loop from bench to bedside is continuous, it's not a one-way journey, and we're continually going back to our patients to see how we can improve things. So with some early support from the BRC and other funders subsequently, we've been able to launch a study called Gene Steps, which is shortening time to evaluation in paediatric epilepsy services. This is an international collaboration with an international partnership, including Toronto, Melbourne, and Boston. And the reason we're doing this is that these conditions can be individually rare but collectively common. Therefore, we want to unite our genomic and paediatric epilepsy expertise. And we are now delivering for patients at Great Ormond Street a 21-day rapid turnaround whole genome sequencing alongside deep phenotyping. This is important because in order to be able to access these novel therapies, we need to make rapid diagnoses. And the kind of novel therapies that we're interested in introducing for the epilepsies are RNA therapies amongst other forms of gene therapies. And I'm currently investigating, along with Haiyan Zhu, an antisense oligonucleotide therapy where a short synthetic RNA molecule could be used to block or increase expression of a certain gene, in this case increasing expression, and this will hopefully lead to more protein and will rescue the phenotype in our patients. And we'll measure this at various different levels to see if we are effective. So I'd like to finish by thanking the GOSH NHR BRC for funding the project, for awarding me the Catalyst Fellowship, which led to generation of the preliminary data leading to my MRC Clinician Scientist Fellowship. This also led to branching into translational readouts, including my TIN award for the ASO development and subsequently supporting the translational therapy development. I'd also like to thank our other funders, but especially GOSH Charity, our partners Young Epilepsy and Rose Trees. And I'd like to finish by thanking my great team that I work with. Thank you.
Thank you for this talk and also all the speakers in this session. And now I want to pass back to Laura.
Thank you very much, Sergi. Just one last thing to wind up with the genomics session, which has been a great session. We wanted to just play you one of the videos that we've developed, the animations that Lynn mentioned around how to explain genomics to a wider audience. So we'll just show you that now.
Our bodies are exceptionally clever. Even when we're asleep, they're doing all sorts of extraordinary things, like mending themselves. These things don't happen by magic. Deep inside our cells is a chemical called DNA. DNA acts like an instruction manual controlling how our body works. These instructions are made up of 4 chemical letters that we call A, G, T, and C. These repeat 6 billion times. That's enough letters to take a whopping 57 years to read out aloud. The complete set that makes you the unique and brilliant individual you are is called your genome sequence. Around 99% of our sequences are the same. It's the 1% that makes us all different, deciding everything from our height to our hair colour. DNA is a bit like the computer code used to give robots instructions. Sometimes a glitch in the code, like a spelling mistake, will create individual differences. Most of the time, this isn't a problem. Sometimes it can even be an advantage. But if a glitch makes the instructions confusing, the robot might not be able to do things as it should. It's the same with our DNA. Everyone has glitches, but some of us have glitches that cause health problems because our body isn't getting the right instructions. For instance, having a glitch in the DNA for your muscles might make you less strong. The only way to find that glitch is to look closely at your genome sequence. Genome sequencing is an exciting new technology. By taking a blood sample, scientists can remove the DNA and read your sequence using a high-powered machine. The scientists will compare your sequence to thousands of other people's to find the glitch. They might even compare it to your close relatives, like your mum and dad. Working out exactly where your genome is different can help doctors to diagnose the cause of your condition and decide on the best treatment. It's still a very new technology, so the more people have their genome sequences read and compared, the more we can learn about DNA and the more answers we can find in the future.